हेलो फ्रेंड्स, आज चैप्टर 14 में एस्टिमेशन ऑफ प्लाज्मा ग्लूकोज एंड ग्लूकोज टॉलरेंस टेस्ट देखेंगे। चलिए शुरू करते हैं। ब्लड ग्लूकोज एस्टिमेशन। द नॉर्मल फास्टिंग ब्लड शुगर रेंजेस बिटवीन 60 to 100 milligram per 100 ml after a carbohydrate meal or glucose drink. Blood sugar level rises quickly and reaches a maximum, usually not more than 60 milligram above the fasting level in about 1 hour time. Then it rapidly returns to normal fasting level or even lower in about 1/2 to 2 hour time. If the blood sugar does not return to near fasting level in 2 hours, and if the maximum level reached show a difference of more than 60 milligram above the fasting level, it indicates low glucose tolerance, as is seen in diabetes mellitus. There are several methods available for estimation of blood glucose level. These are based on two principles. Glucose being a reducing substance, its reducing property is utilized for its estimation. Besides glucose, there are a few other reducing substances such as glutathione and ergothioneine in blood, which are mostly situated inside the red blood cells. If red cells lysis is avoided, any of these techniques give results which are very near to actual glucose level. The estimation of glucose by using glucose oxidase enzyme or hexokinase is increasingly being used by many laboratories. This enzyme oxidates glucose and liberates equivalent amount of hydrogen peroxide, which is estimated. This method is very specific for glucose. The estimation of glucose by using glucose oxidase enzyme or hexokinase is increasingly being used by many laboratories. This enzyme oxidizes glucose and liberates equivalent amount of hydrogen peroxide, which is estimated. This method is very specific for glucose. Two methods are described below. One is a colorimetric method of orthotoluidine and other is enzymatic method using GOD and POD. Prevention of glucose. It must be emphasized that glucose in blood is very labile on account of the presence of glycolytic enzymes. Preservation of blood. It must be emphasized that glucose in blood is very labile on account of the presence of glycolytic enzyme. Glycolytic enzyme से क्या हो सकता है? ग्लूकोज टूट सकता है इन रेड ब्लड सेल्स, जो कि रेड ब्लड सेल में होता है ग्लाइकोलाइटिक एंजाइम. If the blood has to be left for more than a few minutes before starting the estimation, it is necessary that it be collected in a fluoride tube containing EDTA as anticoagulant and sodium fluoride as an anti-glycosylic agent and mixed thoroughly. Plasma glucose estimation by GOD-POD method. Principle. Glucose is oxidized by glucose oxidase GOD into gluconic acid and hydrogen peroxide. So produced oxidizes chromogen 4-amino antipyrine and a phenolic compound, para-hydroxy benzoic acid, in presence of peroxidase POD enzyme into red color compound. The intensity of the red color produced is proportional to glucose concentration and is stable for 2 hours. The color is measured in a photometer at 505 nanometer, 490 to 530 nanometer. ओके, फिर से देख लेते हैं प्रिंसिपल क्या है। तो प्लाज्मा ग्लूकोज एस्टिमेशन बाय जीओडी पीओडी मेथड। प्रिंसिपल कहता है ग्लूकोज इज ऑक्सीडाइज्ड बाय ग्लूकोज ऑक्सीडेज, जिसको शॉर्ट में जीओडी कहेंगे, इनटू ग्लूकोनिक एसिड एंड हाइड्रोजन पेरोक्साइड। हाइड्रोजन पेरोक्साइड जो बन रहा है, हाइड्रोजन पेरोक्साइड सो प्रोड्यूस्ड ऑक्सीडाइजेज क्रोमोजेन, क्रोमोजेन को ऑक्सीडाइज कर देता है। तो क्रोमोजेन में क्या है, तो 4 एमिनो एंटी पायरिन एंड या फेनोलिक कंपाउंड पारा हाइड्रोक्सी बेंजोइक एसिड, पारा हाइड्रोक्सी बेंजोइक एसिड है और 4 एमिनो एंटी पायरिन है। तो ये क्रोमोजेन ऑक्सीडाइज होगा इन प्रेजेंस ऑफ पेरोक्सीडेज पीओडी एंजाइम इनटू रेड कलर कंपाउंड। The intensity of the red color produced is proportional to glucose concentration and is stable for 2 hours. The color is measured in a photometer at 505 nanometer, 490 से 530 नैनोमीटर के बीच में। ग्लूकोज O2 GOD ग्लूकोनिक एसिड बना प्लस H2O2 बना हाइड्रोजन पेरोक्साइड। अब H2O2 जो है 4 एमिनो एंटी पायरिन प्लस पारा हाइड्रोक्सी बेंजोइक एसिड के साथ रिएक्ट किया और पीओडी पेरोक्सीडेज एंजाइम से हेल्प से रेड कंपाउंड बना और वाटर बना 2H2O। रिएजेंट्स में क्या क्या रहता है, तो बफर, रिएजेंट 1 में बफर रहता है, जीओडी पीओडी और क्रोमोजेन। फॉस्फेट बफर पीएच 7.4 95 मिलीमोल पर लीटर। ग्लूकोज ऑक्सीडेज 5000 इंटरनेशनल यूनिट पर लीटर और मोल, पर ऑक्सीडेज 5000 इंटरनेशनल यूनिट पर लीटर और मोल। फिर 4 एमिनो एंटी पायरिन 0.2 मिलीमोल पर लीटर, पारा हाइड्रोक्सी बेंजोइक एसिड 5.9 मिलीमोल पर लीटर। रिएजेंट 2 ग्लूकोज स्टैंडर्ड 100 मिलीग्राम पर डेसीलीटर। ग्लूकोज अनहाइड्रस वेल ड्राइड 1 ग्राम पर लीटर। These reagents can be obtained as ready-to-use kit from commercial chemical company. Procedure क्या है, तो 3 small test tube BST for blank standard and test respectively. Blank में, स्टैंडर्ड में और टेस्ट में सब में GOD POD reagent 1 ml लेना है। स्टैंडर्ड सॉल्यूशन में 10 माइक्रोलीटर स्टैंडर्ड डालना है और टेस्ट में 10 माइक्रोलीटर सैंपल डालना है प्लाज्मा। Incubate all tubes at 37 degrees Celsius for 15 minutes. Mix and read OD at 505 nanometer. Transmission in photometer using 1 ml. Cubate after making 0 adjustment with the blank. Glucose concentration in milligram per 100 ml serum. Glucose concentration in milligram per 100 ml serum कैसे कैलकुलेट करेंगे, तो OD of tube T, OD of tube S into concentration of sugar in standard, that is 100 milligram. Note क्या करना है, the method is specific because enzyme glucose oxidase acts only on glucose. Therefore, this method gives true value of blood glucose. Calibration graph. Calibration graph shows reading of assay against rising concentration of analytes. For example, you can determine the concentration of glucose from graph prepared by you by recording optical densities reading OD of glucose estimation at different concentration. For example, at 50, 100, 200, 300, 400 and 500 milligram per 100 ml concentration of glucose in a set of samples. After preparing the graph, you will notice that with method adopted by you, the graph goes straight up and then bends down at certain concentration. The concentration of analyte during which the graph goes in a straight line indicates the useful range of the method. If a sample has higher concentration beyond the useful range, then the sample has to be suitably diluted. Finding out concentration of an analyte using a particular method is easy by looking at the graph, but graph has its limitation because of many uncertainties such as electrical variation, temperature of the environment, deterioration of in instrument function with time. Therefore, one graph should not be used for all time to come. It has to be prepared at intervals of few months. An example of preparing a calibration graph for glucose using GOD/POD method is given below. A stock glucose solution containing 1000 mg per 100 ml is prepared. Glucose should be dried in a desiccator for 24 hours before using it. Then solutions having different concentration of glucose are prepared in as given below. सैंपल 1 से लेकर 7 तक. वॉल्यूम ऑफ डिस्टिल्ड वाटर 1.9 से लेकर 0.8 तक. वॉल्यूम ऑफ स्टॉक ग्लूकोज सॉल्यूशन 1 से लेकर बढ़ते हुए 1.2 तक. फाइनल ग्लूकोज कंसंट्रेशन 1 लिए तो 50, 100 मिलीग्राम, 50 मिलीग्राम, 100 मिलीग्राम, 200 मिलीग्राम, 300 मिलीग्राम, 400 मिलीग्राम, 100 milligram, 600 milligram. Mix well all solution. Now find out optical densities of each of the above glucose solution estimated by using GOD/POD method for glucose estimation. Do not forget to keep one test tube for blank. Then plot your graph keeping OD in ordinate and glucose concentration in abscissa. Note down your observation for the useful limits of GOD/POD method for determining glucose concentration in any sample including blood. कुछ क्वेश्चंस हैं. पहला है why a straight line in graph bend at certain glucose concentration. Answer is Lambert-Beer's law states that OD of the solution will be proportional to the product of the concentration of color substance and the depth of the liquid in the cuvette through which light passes. If the depth of liquid is kept constant by taking same or similar cuvette, then OD will be proportional to the concentration of colored substance within certain limits, beyond which proportionality fails. While same calibration graph cannot be used for all time to come because the color reaction which was taken at a certain temperature of the day will not be the same when weather changes or instrument ages and electrical variation takes place. Therefore, the calibration graph should be checked at frequent intervals. Orthotoluidine method. Principle: glucose condenses with orthotoluidine in glacial acetic acid. when heated at 100 degrees Celsius. The product is a blue-green and glucose amine. The color so produced is measured at 630 nanometer and is proportional to the amount of glucose percent. Reagent क्या क्या रहता है तो benzoic acid solution 0.1% in water. This is stable indefinitely at 50 to 25 degrees Celsius. A stock glucose standard solution 1000 mg per deciliter. Dissolve 1 gram of pure and dry glucose in 100 ml of 0.1% benzoic acid solution. Store frozen in a small vial. Under this condition, it is stable indefinitely. Dissolve 1 gram of pure and dry glucose in 100 ml of 0.1% benzoic acid solution. Dissolve 1 gram of pure and dry glucose in 100 ml of 0.1% solution. Store frozen in a small vials. Under this condition, it is stable indefinitely. Working glucose standard 100, 200, 400 mg per 100 ml. Allow a stock glucose solution to attain room temperature. Dilute 10 to 20, 40 ml of stock to 100 ml with 0.1% of benzoic acid solution. These will keep well for 2 months or more at 20 to 25 degrees Celsius. Orthotolerin reagent. Dissolve 1.5 gram thiourea in about 960 ml of glacial acetic acid annular. When completely dissolved, add 60 ml orthotolerin. Mix well. पहले थायोयूरिया 1.5 ग्राम जो है 960 ml ऑफ ग्लेशियल एसिटिक में डिजोल्व कर लिए. उसके बाद जब कम्प्लीटली डिजोल्व हो गया तो उसमें 60 ml ऑर्थोटोलिन डाल दिया. मिक्स वेल एंड स्टोर इन योर ब्राउन बॉटल. द रिएजेंट इज स्टेबल फॉर अप टू 3 months एट 20 to 25 degrees Celsius. Note: orthotoluene is highly corrosive and hence it should not be pipetted by mouth. Orthotoluene reagent should not be used immediately after preparation. Use it 24 hours after preparation. Make 3 ml toluene reagent in each test tube for test standard and blank. And transfer 0.1 ml of plasma, standard and distilled water into respective tube. Mix well and put them in boiling water bath for exactly 12 minutes. At the end of boiling, remove the tube and cool them in cold water. Read the absorbance of each tube in a photoelectric colorimeter. Using a red filter 630 nanometer transmissions, the color is stable for at least 1 hour. The cuvette should be dry for reading the absorbance. Calculation: milligram glucose per mg glucose per 100 ml serum. T minus BS minus B into mg glucose in 100 ml standard. Note: plasma or serum but not whole blood can be used for measuring glucose in blood. Avoid hemolyzed serum, plasma or serum, cerebrospinal fluid. Sugar can also be measured by this method. क्वेश्चन है कुछ देख लेते हैं. वाई इज अ फ्लोराइड ट्यूब यूज्ड फॉर कलेक्टिंग सैंपल फॉर ब्लड शुगर एस्टिमेशन. तो ग्लूकोज प्रेजेंट इन द ब्लड सैंपल कलेक्टेड फॉर एस्टिमेशन undergo glycolysis by RBCs so that sugar value decreased by 5 to 10 milligram per deciliter per hour. Fluoride prevents this by inhibiting the glycolytic enzyme enolase. Question number 2. What do you understand by fasting, postprandial and random blood sugar sample? Fasting sample is taken after an overnight fast, nothing being taken by mouth in the morning except water. Postprandial sample is taken 2 hours after meal or 75 gram of glucose intake. No other food is taken within that time. Random sample is taken at any time irrespective of when the last meal was taken. Question number 3. What is the normal blood glucose values? Random blood sugar less than 160 milligram per deciliter. Fasting blood sugar less than or range between 60 to 105 milligram per deciliter. Postprandial less than 130 milligram per deciliter. Name the hormone which regulates blood sugar level in body. Insulin decreases the blood sugar level by increasing glucose utilization by tissue. Glucagon, growth hormone. Thyroxine, corticosteroids increases the blood sugar values. What is insulin? Insulin is a 34 amino acid polypeptide hormone which is secreted by the beta cells of the islet of Langerhans in the pancreas in response to hyperglycemia. What is the mechanism of insulin released from the beta cell? The beta cells are freely permeable to glucose via the glucose 2 transporter and the glucose is phosphorylated by glucokinase. Therefore, increase in blood glucose increase. Therefore, increase in blood glucose increases metabolic flux through glycolysis, the citric acid cycle, and the generation of ATP. The increase in ATP concentration inhibits ATP sensitive K potassium channel, causing depolarization of the cell membrane. This increases calcium 2 plus influx via voltage sensitive calcium 2 plus channel, stimulating exocytosis of insulin. Thus, the concentration of insulin in blood parallels that of the blood glucose. Question number 7. Name some substance which cause release of insulin from pancreas. Apart from glucose, other substances which can cause release of insulin from pancreas includes amino acid, fatty acid, free fatty acid, ketone bodies, glucagon, secretin and sulfonylurea drugs, tolbutamide and glyburide. What are the causes of hyperglycemia? Diabetes mellitus, hyperactivity of pituitary, thyroid or adrenal glands. Drug-induced prolonged use of steroids. How is the blood sugar level regulated? Insulin is the only hormone that is secreted in response to hyperglycemia and it lowers blood sugar level by increasing the peripheral utilization of glucose. It recruits GLUT receptor to promote glucose transport into the cells and increases its utilization in glycolysis and glycogenesis. It inhibits gluconeogenesis and glycogenolysis. In hypoglycemia, glucagon is the main hormone that is secreted. It increases the blood sugar level by enhancing liver glycogenolysis, glyconeogenesis, and inhibiting glycolysis. What is the cause of hypoglycemia? Hypoglycemia, where the blood sugar is less than 60 mg. Common cause: overdose of insulin and other sugar-lowering drugs using during treatment. Iatrogenic cause: hypothyroidism, glucagon storage disease, insulinomas. What is true glucose value? Blood glucose, which is estimated using enzymatic method GOT/POT, is called true glucose value because this enzyme only acts on glucose and no other reducing agents. Ortho-Toledo method uses the reducing property of glucose for its estimation. There are other non-glucose reducing agents in blood which will affect the result. The other non-glucose reducing agents are ascorbic acids, glutathione from hemolyzed RBC, uric acid, etc., which also react with ortho-Toledo, giving a higher blood sugar values. Blood sugar, which is estimated using enzymatic method GOT/POT, is called true glucose values because this enzyme only acts on glucose and no other reducing agent. The ortho-Toledo method uses the reducing property of glucose for its estimation. There are other non-glucose reducing agents in the blood which will affect the result. Thus, the other non-glucose reducing agents are ascorbic acid, glutathione from hemolyzed RBC, uric acid, etc., which can react with ortho-Toledo, giving the higher blood sugar values. Glucose tolerance test. Glucose tolerance test (GTT) is performed on a person to find out how this blood glucose responds to a test dose of glucose orally. The test is done in fasting condition on an individual who has been on normal diet in the last 72 hours. A sample of blood and urine is collected in fasting state, and then the person is given a standard dose of 75 gram or 1 gram per kg body weight of anhydrous glucose in 300 ml water to drink. The time is noted. Subsequently, blood is collected every half hour and urine every hour till 2 hours. Glucose is determined in all 5 blood samples, and test for presence of glucose is done on all 3 samples of urine. In normal individual fasting, blood glucose varies between 60 to 104 mg per 100 ml. The glucose level starts rising soon after glucose drinks and reaches a peak within 25 to 60 minutes, which is not more than 60 mg above the fasting level in normal individual. Then the level start falling and come back to fasting level or even below by 90 to 120 minutes. This is described as normal glucose tolerance. When the fasting glucose level is above 120 mg/cent and maximum level attained is above 200 mg/cent, or if the level does not return to fasting level within 2 hours with or without glycosuria, the person is said to have lower glucose tolerance, that is diabetes mellitus. Renal threshold, normal, GTT, diabetes, GTT, lag curve. In normal person, glucose is not detected in any of the above three urine sample. If glucose is present in one or all urine sample, it means that the blood glucose level in the individual had been above the renal threshold of glucose. In this way, one can find out renal threshold of a person. Renal threshold in normal people lies between 160 to 200 milligram percent glucose, 180 in blood. In some individual it may be below this level due to renal defect, which will result in glycosuria. Even if the blood glucose had been within normal range of GTT, this is known as renal glycosuria, and the person is suffering from impaired glucose tolerance. Renal threshold is higher in certain renal disease, and such patient may not show glucose in urine, although blood glucose level indicates diabetes mellitus. Renal threshold in normal people lie within 160 to 200 milligram glucose, 180 in blood. In some individual it may be below this level due to renal defect. In certain individual, level of blood glucose during GTT goes very high. If above renal threshold between 30 to 60 minutes after glucose ingestion, but the blood glucose level drops down to normal level in 90 minutes. The GTT curve in this type of individual is known as lag curve. This is because there is delay in secretion of insulin, but insulin secretion quickly picks up and the glucose level comes down to normal within 90 to 120 minutes. Such individual may also show presence of glucose in urine sample taken in 60 minutes. Such individuals are not diabetic and need not worry about it. In few persons, 30 to 60 minutes after glucose ingestion of sugar level in blood rises just slightly above the fasting level, that is above about 20 to 30 milligram. This happens when there is a defect in absorption, as in celiac disease, or due to poor secretions from thyroid, pituitary and adrenal cortex. Such curve is known as flat curve. Questions and answer. Name the different types of glycosuria. Name the different types of glycosuria: alimentary glycosuria, renal glycosuria, diabetes glycosuria. What do you understand by the renal glycosuria and enumerate its cause? Condition characterized by urinary excretion of glucose at blood concentration of glucose below the renal threshold value. Causes defect in sodium glucose co-transporter SGL2 in proximal tubules. Pregnancy due to reduction in the tubular reabsorption capacity. What do you understand by the term elementary glycosuria and enumerates its cause? Condition in which fasting and 2-hour values are normal, but an exaggerated rise in blood glucose level in between above the threshold value, leading to glycosuria following the ingestion of glucose or meals, in partial total gastrectomy, and in hyperthyroidism. What do you understand by the term renal threshold? What is the renal threshold for glucose? Renal threshold is the highest level of substance in blood up to which it can not appear in urine. Renal threshold of glucose is 180 ml per deciliter. This means that glucose starts to appear in urine when its blood level is more than 180 milligram per deciliter. ओके, इस चैप्टर कंप्लीट हो गया। नेक्स्ट चैप्टर है 15 पॉइंट ऑफ केयर टेस्टिंग, POCT, ग्लूकोमीटर। चलिए इसको नेक्स्ट वीडियो में कवर करेंगे। तब तक के लिए बाय बाय। हंसते रहिए, मुस्कुराते रहिए और पढ़ाई भी करते रहिए। बाय बाय।